Hello everybody, and welcome back to BioSC 140, Human Physiology. This is the S2P3 Nervous System Part 2 video. In this video, we're going to cover the different cells and some characteristics of them within the nervous system. So, we have the nervous system as a whole. There's two main types of cell within the nervous system. There's the neuron. The neuron is the famous one. The neuron is the one that sends those electrical signals over a long distance. The neuron is the one that has the computing power, those interneurons in our central nervous system that help you think. When people think of what the nervous system does, they're thinking of neurons. It's the functional component of the nervous system. Those long distance electrical signals, that computing power of our mind. Glial cells are very, very important also, but you don't hear about them too much in just our everyday lives. Glial cells are the support cells. Neurons are highly adapted. They need extra help in order to do what they do. And they have this group of cells called glial cells. There's different types of glial cells, but these glial cells are the supportive cells. Their job is to keep the neurons happy. Their job is to assist the neurons in doing what the neurons do. And glial cells are very, very important. Also, there are many disease processes that arise from issues with glial cells. So they're very important also in healthcare. Glial cells provide physical and metabolic support for neurons. Glial or neuroglia are the support cells. And of the glial cells, there are two main types that you'll hear about. You'll hear about oligodendrocytes and Schwann cells. Oligodendrocytes are in the central nervous system. Oligodendrocyte, central nervous system. Schwann cell, peripheral nervous system. Peripheral nervous system. Oligodendrocytes and Schwann cells, on top of providing physical and metabolic support, help to produce myelin. Myelin are these round insulating things on the axons, and myelin is super, super important to the function of neurons. There are many diseases that affect myelin that have devastating effects on a person's life. So, central nervous system, myelin comes from oligodendrocytes. Peripheral nervous system, myelin comes from Schwann cells. You can see here, to the left of this spot is central nervous system. To the right is peripheral nervous system. You got oligodendrocytes here and Schwann cells here. So myelin, what is myelin? If I say it's so important, and if myelin is informed properly, you have diseased states. Well, myelin is actually concentric layers of cell membrane. If you look at this microscope photo here, you can see the axon in the middle, and it looks like an onion. It's all these layers. It's all these extra lipid layers. It's extra layers of cell membrane, of lipid membrane. It provides structural support, and it speeds up the rate at which signals are sent, which action potentials are sent. You'll see that the myelin here is on the axons. You don't see the cell bodies wrapped up in myelin. You see the axons wrapped up in myelin. And so it's structural support for the axon, and it helps speed up the speed at which that signal is sent. So myelin, concentric layers. So I'm going to tell you kind of a weird story that's going to help you remember what myelin is. So You might have heard of like gray matter and white matter in someone's brain or in like the central nervous system, you know, the gray matter of the brain, the white matter of the brain. When you see or hear about gray matter in the central nervous system, gray matter is primarily made up of cell bodies. White matter is primarily made up of axons surrounded in myelin. Axons surrounded in myelin. So axons surrounded in myelin, white matter, cell bodies, gray matter. So what is myelin made up of? It's concentric layers of cell membrane. It's phospholipids, lipids. In some areas around the world, it's common to eat brains, like sheep brains. I remember when I was taking neurobiology class and I went to the dentist. And my dentist was from Southeast Asia. And, you know, as he's working in my mouth, he's like, as he's, you know, working on my teeth, he does some small talk. And he tells me, and I tell him, you know, I'm in neurobiology class. And he starts talking, oh, I love that class, this. And then he goes, have you ever eaten brain before? And I said, no. He goes, in my country, we eat brains. We eat sheep brain. And he's like, do you know what it tastes like? And I was like, I have no idea why my dentist is talking about eating brains right now while fixing my teeth. And he goes, brain is just like a big hunk of fat. He's like, you know how when you eat a steak and there's like that piece of white fat on like the side of the steak? He's like, that's what brain's like. Brain's like that piece of white fat. It's just a big gooey ball of fat. And all that fat comes from myelin. It's all those myelin sheets, all those lipid layers around the axons, all that white matter. Lipids. All right. All right, so back to the slides. Glial cells, oligodendrocytes in the central nervous system, Schwann cells in the peripheral nervous system. In the central nervous system, it's usually about one oligodendrocyte for every axon. So one axon, one oligodendrocyte. In the peripheral nervous system, where you have Schwann cells, there's usually hundreds of Schwann cells for every axon, 500 or so, hundreds, it varies, but hundreds of Schwann cells per axon. Who is this person right here? That man played Superman in the old Superman movie. His name's Christopher Reeve. And he's famous for being Superman, being a good actor, and he fell off his horse and broke his spinal cord, injured his spinal cord. And he was paralyzed for the rest of his life. He couldn't use his legs. The axons, the cells, so the cells and the axons in his spinal cord, in his central nervous system, spinal cord, central nervous system, never regenerated. It's thought that the inability or the central nervous system, you don't really regenerate axons well. Some argue it's possible, but everyone agrees it's not very good for regeneration of damaged axons in the central nervous system. So he never got his legs back, or his function of his legs back. This guy right here, because it was central nervous system damage. This guy right here, Jesse Arbogast, he was a little boy, and he was playing in the ocean, and he got his leg bit by a shark. I'm not sure if it was bit completely off or just partially off or what exactly how it was, but he got bit by a shark. And doctors put his leg back together, like cooked it back into his body, and he was able to have a functioning leg after it. I think it got completely bit off. Like, I'm pretty sure the shark completely bit his leg off, but I'd have to check the story. It's been a few years since I've checked up on the story. But yeah, leg either got bit off or almost bit off. Doctors put it back together. The axons regenerated. axons regenerate. It's thought that peripheral nervous system axons regenerate better because of this large number of Schwann cells to axon ratio. So a nerve, when we talk about a nerve is a bundle of neuronal axons. So we have the cell body, and then we have this axon, which is the long arm that sends this action potential, that sends this electrical signal. We saw this with the frog lab. Nerves are bundles of axons. Some nerves are only motor nerve, only contain efferent fibers. So they're motor nerves, they only contain efferent fibers. Some only contain sensory. Lateral femoral cutaneous nerve is only sensory, it only has afferent nerves. Some nerves are mixed nerves. Mixed nerves. They contain afferent and efferent fibers. The sciatic nerve contains both afferent and efferent fibers. So a nerve is a bundle of neuronal axons in the peripheral nervous system. Peripheral axons can regenerate after damage. So this is Jesse Arbogast. This is the shark victim. Peripheral nervous system axons can heal. There's a big process that happens, but the glial cells are very important for this process. So lateral femoral cutaneous nerve is sensory only. If you damage that nerve, what are the signs and symptoms? What are the signs and symptoms of damage to a sensory only nerve? Well, it'd be numbness. It'd be lack of sensation because it's a sensory nerve. If you damage a motor nerve, what are the signs and symptoms? Lack of motor function. The sciatic nerve is a mixed nerve. It's a mixed nerve. Most people have heard of sciatica or sciatic nerve. What if your sciatic nerve just stopped functioning completely? What would be some signs and symptoms? Complete does not work at all. Well, numbness, because there's sensory fibers, and paralysis, not able to activate your muscles because there's efferent motor neurons also. Neurons cannot replicate. They don't undergo mitosis. So once you're an adult, like the nerves you have are the nerves you have. Peripheral nerve axons can regenerate, but cell bodies can't. It's the axons that are regenerating, not the cell bodies. Schwann cells produce chemical factors that signal damage has occurred and keep the cell body alive to stimulate growth. There's a whole process. Like if this nerve was cut right here, the Schwann cells would start to kind of eat the nerve back to this Neuron VA and eat this nerve back to this Neuron VA. And then after it's kind of clean up the area and start regenerating a new one. There's a whole process. But basically the Schwann cells kind of take care of the cell. They keep it alive and they stimulate that healing process. Yeah, proximal stump, distal stump. Yeah, there's a whole process. Perfect. So neuron ratio influences axonal regeneration. Perfect. All right, so neurons are nervous system cells with three main parts. I've used a lot of this terminology already, but let's get into it. Cell body. The cell body is pretty self-explanatory. It's where the nucleus is. It's where all those organelles are. It's where all that machinery is. It's the integrative center. It's the processing center. It's the factory for proteins. It's the factory in the brains. Dendrites are these little arms coming off the cell body, and they receive input. Other nerve cells send signals to dendrites. Dendrites receive signals, receive inputs. The axon is what people think of when they think of what the nervous system does. It's the one that sends that action potential. It's the one that sends that signal, the axon. A few other words, the synapse So you can see right here, this axon is synapsing or forming a synapse with this dendrite right here and with the cell body right here. The synapse is the place in between the synapse, the junction or small space between the axon and the target cell, axon and target cell. So presynaptic, before the synapse cell, is the one that sends the signal. So in this situation, is it the left or the right cell? That's the presynaptic cell. It's the one sending the signal. It's this one. This one has the axon sending the signal to the synapse. It's the presynaptic cell. The postsynaptic cell is the one receiving the signal. So this one is, on the receiving end of the signal, this cell down here, postsynaptic cell. And postsynaptic or presynaptic is always going to be a nerve. Postsynaptics could be a neuron, a muscle, or a gland. The neurotransmitter are these chemical messengers that go across the synapse. So in most situations, we're going to talk about the exception, but in most situations, this cell decides it wants to send a signal. It's going to send that signal down the axon in the form of an electrical signal. That electrical signal at the synapse is going to get turned into a chemical signal via neurotransmitters. Neurotransmitters are going to get released from the synapse, and it's going to carry that signal to the next cell over, the postsynaptic cell. And then in this case, that postsynaptic cell turns that signal back into an electrical signal, sends it down the axon. Neurons can be characterized by structure and function. So different ways of classification. On the first day of class, we talked about functional and structural names for different organ systems. So right here, we have functional names for different neurons. So afferent. Afferent is sensory. It carries information towards the central nervous system. So any sensory nerve, any nerve that carries sensory information to the central nervous system, afferent, functional name. Interneuron relays information within the central nervous system. These are your computing neurons, your processing neurons. Any neuron that relays information within the CNS, interneuron. Efferent motor, functional name again, carries information away from the central nervous system to an effector. Anything that carries information away from the central nervous system to something that can affect change, efferent nerve. Functional names. Afferent, functional name, interneuron, functional name, efferent, functional name. Neurons can also be characterized by their structural name. So we've got pseudo-unipolar, bipolar, and multipolar. So let's start with multipolar, actually. Multipolar is the easiest. It has multiple dendrites. Multiple dendrites. Look at this. Lots and lots of dendrites receiving input, one axon with an output. Lots and lots of dendrites receiving inputs, one axon output. Multipolar are often central nervous system interneurons as well as efferent motor neurons. Bipolar, single axon, single dendrite, single axon, single dendrite. So right here we have single dendrite, single axon. You're going to find these in your special sensitives. So smell and vision. Smell and vision is where you're going to find your bipolars. Your pseudo-unipolars are most of your other sensitives, hot, cold, touch. And you're going to have a cell body with one process that comes off it, but that one process kind of looks like two processes. Pseudo-unipolar. It's got some dendrites. It's got the axon. That's most other senses. So this is what I was talking about before. Your presynaptic cell is going to send an electrical signal down the axon. When it gets to the end of that axon to the synapse, it's going to turn that electrical signal into a chemical signal. Those neurotransmitters are going to be released into the synaptic cleft or the synapse. Those neurotransmitters are going to diffuse across the synaptic cleft and interact with receptors on the other side on the postsynaptic cell and cause a change on the postsynaptic cell. The synapse is the point where the axon meets the target. You can have a varied number. Some cells can have hundreds of different synapses. Some only have a few, some will have hundreds, literally hundreds of synapses. In fact, there's a lab coming up where we see an electron microscope photo of a cell body that's synapsing with like 100 different synapses or axons. Some cells have many dendrites, so they can receive a lot of different inputs. There can be synapses all over all these dendrites and the cell body. Presynaptic cell releases the signal, so that's the one that has the electrical signal that's turned into a chemical signal. Postsynaptic receives the input. It has the receptor for the neurotransmitter. Signal receptor and anatomical connection provide specificity. So that's how most synapses work. It's that electrical to chemical to electrical situation. That's how most of them work. There are a few exceptions that I'm only going to talk about all class in like this line right here and one more slide. That's it. This slide right here and this line right here, because they're not common. They're not really clinically applicable. But there's two types of synapses. There's electrical synapse. They're rapid, but you have less control. That's not what I've been talking about so far. And then there's chemical synapses, which are slower, but you have more control. And that is what I've been talking about so far. So all the synapses I've talked about so far have been chemical. They're the most common. They're slower, but you have more control. When people talk about synapse, this is what they're talking about. Electric, you don't hear about much at all. They're not really clinically applicable. But it's important to know that they're there. So electrical synapses, this is where you have a gap junction. This is where you have a connexon. This is where you have direct connection from one cell's cytoplasm to the next. And it allows the electrical activity of one cell to pass directly into the next cell very fast. It's good because it enables synchronized activity. The electrical activity goes right through synchronized activity. It's not good because that signal can go both directions. There's not control. Signal can go in both directions. There's not really control. You're only really going to find these in your central nervous system. Most situations, it goes electrical signal to chemical signal. That's the release of neurotransmitters. Then chemical signal received by the receptor on the postsynaptic cell will either cause an electrical signal to be produced or a chemical signal to be produced. It's slower, but it's still very fast. I mean, this is the majority of synapses in your body. I mean, think about how fast you think and move and control all the, we're still really, really fast to our perception. Increased control and specificity. It's one directional. You can't have a signal go the wrong direction, go both directions with this setup. So you can also, it gives you another area where you can control. You can alter this synapse. You can control the pathway in more ways via this synapse. A lot of medications work at the synaptic level. SSRI, selective serotonin reuptake inhibitors. There's lots of medications that affect the synapse. Regulate signal production, degradation, or receptor activity. So you're able to control it because you can regulate what happens to these neurotransmitters. You can regulate the production and degradation of neurotransmitters. You can also regulate receptor activity. So orthodromic is one directional, right? It actually means like straight direction. Ortho means straight, like orthodontist is like ortho is like straight. Orthoptera is actually the scientific name for grasshoppers because grasshoppers have straight wings. So cell body to axon helps prevent enterodromic. So enterodromic is what you can get up here where it's the wrong direction, going backwards. With electrical synapses, you can get both directions. With chemical synapses, you only get one direction. All right. So, what are different ways that you can control the signal at the synapse level? Well, you can affect active removal. So you can so this cell releases the neurotransmitter. Then what's going to happen is that signal always going to be there. Are those neurotransmitters always going to be in that synaptic cleft? Is that signal never going to get turned off? Well, you can regulate how you turn that signal off. Active removal is using energy, ATP, to physically pump those neurotransmitters away. Active degradation, so right here's an enzyme that breaks that neurotransmitter down. That's what happens at our neuromuscular junctions. So where our nerves meet our muscles, those neurotransmitters get broken down by enzymes. Diffusion, sometimes those neurotransmitters just need to diffuse away. Target receptor downregulation. So we've talked about that in the communication. Change the number of receptors on the opposite side. Right here is what's going on at your neuromuscular junction. Acetylcholine is released. It binds onto the receptor. Acetylcholinesterase breaks that neurotransmitter into choline and acetate. Boom. Boom. The signal's broken down because you've chopped that neurotransmitter in half. You can actually then reuptake those individual parts into the cell and make more acetylcholine, so you don't waste those parts. You can degrade the signal and then recycle those parts. Norepinephrine is also recycled in a similar way. Neurotransmitters are really important to healthcare and really important to healthcare in general, because there's so many drugs and so many medications that affect neurotransmitters. All of these that you see right here, they all affect neurotransmitters, whether they're medications or recreational use. They all affect neurotransmitters, and that just shows you how big of an effect medications can have on this process right here, on the process of neurotransmitters. All right, everybody, I'm going to stop part 2 right here. I will see you in the next video. Email me if there's anything at all that I can do for you. Have a great day.